Today I will be talking about my research on a putative conjoined sulfate A degrading operon from Tannerella forsythia. The reason I'm studying this protein is because of periodontal disease, which is an infection of the tissues surrounding and holding onto teeth, including gums, periodontal ligament, and alveolar bone. The early stages of periodontal disease, called gingivitis, is characterized by swelling, redness, and light bleeding. As the disease progresses towards more advanced forms, called periodontitis, the gum line starts to recede, even causing bone deterioration in aggressive periodontitis and tissue death in necrotizing periodontitis. The disease is caused by polymicrobial infection, with over 700 known species to infect the periodontal tissue. These bacteria form biofilms that assist the microbes in adhesion, antibiotic and chemical resistance, as well as metabolite cross-feeding. Spearheading the infection is a group of bacteria called the red complex, which consists of Treponema denticola, Porphyromonas gingivalis, and Tannerella forsythia. Tannerella forsythia is a gram-negative anaerobic bacterium and is the only member of its genus. It expresses a number of virulence factors such as glycosidases and is believed to contain many other glycosidic pathways as well. Using genomic analysis, this was the structure for an operon for one of those predicted glycosidic pathways and the predicted functions of each protein. BFO 3043 is a hybrid sensor histidine kinase response regulator that was discovered using string analysis to co-occur with BFO 2285, the first gene in the operon. Using BPROM, it was found that there is likely a second promoter region between BFO 2287 and BFO 2288, with currently unknown associated binding proteins. I've constructed a model of the system based on similar systems found from Bacteroides theta iodomicron, as well as by using psort to predict the localization of some of the proteins, and my research focuses on the early stages of the system. The chondroitin sulfate polymer is likely to be digested by the lyase, breaking it into smaller pieces with the assistance of a surface glycan binding protein, which is then shuttled as disaccharides through the outer membrane into the periplasm, then digested by BFO 2290, the sulfatase, as well as BFO 2285, which is a GHED8 family glucuron hydrolase, before being shuttled through by a major facilitator transporter into the cytosol for further degradation. So this brings me to my research objective. My goal is to study the structure and function of these three proteins, characterize the products, as well as sort of elucidate the pathway, and also use transcriptomics with the RT-PCR to see if these genes are actually upregulated. My hypothesis is that BFO 2290 is the 4-aryl sulfatase that acts after BFO 2291, but is a critical step before BFO 2285 is able to hydrolyze the glycosidic bond between the two monosaccharides. So the first step was recombinant expression and purification. For BFO 2291, it was pretty easy, just recombinantly expressed in E. coli, then use nickel NTA, immobilized metal affinity chromatography, and then I desalted it and concentrated, and here you can see the dynamic light scattering profile of the purification, with the majority of the protein in what appears to be a folded and unaggregated conformation. This protein was solubilized using 30 millimolar sodium phosphate at pH 8 and 50 millimolar sodium chloride. BFO 2290 was purified in pretty much the same way, except using HEPs instead of sodium phosphate. And there were actually two constructs that we used, because originally when we recombinantly expressed BFO 2290, there wasn't a match arrays to make the protein active, and so we expressed it in a PET duet with another protein, BFO 1635, which produced an active protein, which henceforth is going to be called BFO 2290+. Again, a lot of the protein after purification was what appeared to be in a folded state. BFO 2285 was unable to be purified using immobilized metal affinity chromatography, which I suspect is because it's localized in the periplasm, and usually those proteins have chaperone proteins that help it fold. So I used ammonium sulfate precipitation instead and followed it up with a secondary purification using anion exchange, although the dynamic light scattering data would indicate that it is, for the most part, folded in some stable conformation. I use these purified proteins to perform fluorophore-assisted carbohydrate electrophoresis, in which a fluorophore, in this case 2-aminoacridone, is bound to the carbohydrate and then run through a high percentage acrylamide gel. The control labeled A is just chondroitin sulfate that was dissolved and then run through the gel. Then B, C, and D were in order single protein digests of the carbohydrate using BFO2291, BFO2290, and BFO2285. As expected, only BFO2291, the lyase, was able to make a substantial difference in the banding pattern. Next was the double protein digests, E, F, and G. In E, we can see that again there was a change in banding pattern with BFO2291 and 2290. BFO2291 and 85 produced no significant change from B, which was just BFO2291, and G, using BFO2290 and 2285, produced the same banding pattern as the control. This would agree with my hypothesis and suggest that BFO2291 and 90 act sequentially. Finally, there was the triple protein digest in H, where I was expecting to find another different banding pattern, but what I found was pretty much identical to E, which was just the digest of BFO2291 and BFO2290, which would indicate that BFO2285 was either inactive or unable to perform its catalytic function in this state. I also did an H1 NMR of the digest products of BFO2291 as well as BFO2290 after being digested with BFO2291 and saw a shift in peaks that suggested that there definitely was a different product being generated each time compared to the standard. Unfortunately, I can't show the NMR spectrums as I currently don't have access to them. I then proceeded to do kinetic assays with BFO2285 and BFO2290, and as I suspected, BFO2285 was not active. For BFO2285, I was looking for a decrease in absorbance at 232 nanometers caused by the unsaturated product of BFO2291, and with both just the digest of BFO2291 as well as the double digest using the sulfatase, I saw no change in absorbance over the course of an hour. For BFO2290, I used a pH-dependent assay because paranitrophenol sulfate and paranitryl catechol sulfate were not active with BFO2290. Of course, for this assay, I was using BFO2290 plus, which is the version that was expressed in the PET duet with the post-translational modification enzyme. So looking at the mechanisms on the bottom right, the exact mechanism is kind of disputed amongst the sulfatase community, but we do know that hydronium ion is released in both cases. So the whole assay was run at extremely low buffering conditions using p-nitrophenol as a colorimetric pH indicator at 405 nanometers. Since I was using p-nitrophenol as the indicator and it has a pKa of 7.14, I had to switch to MOPS buffer instead of HEPES, which has a closer pKa than HEPES does. And as the reaction proceeds, I'm expecting a drop in pH and thus a drop in absorbance at 405 nanometers. And that's exactly what I saw, which agrees with the phase gel that I ran earlier. I converted the absorbance to concentration using a standard curve that I titrated with hydrochloric acid and produced the Michaelis-Menten curves on the right. Looking at the top graph, we can see that using the digested product of BFO2291 as the substrate for this reaction, we eventually reach Vmax, and it's a nice Michaelis-Menten kinetic curve. But using undigested chondroitin sulfate, it's a straight line and never quite reaches Vmax under the same chondroitin concentrations, which would indicate that BFO2290 is an exo-acting enzyme, meaning it will only cleave the terminal end or the completely digested disaccharide product of BFO2291. And the kinetic parameters that are reported here are specific to this purification and this version of BFO2290 because there's no way of actually determining what percentage of the purification was active protein versus inactive protein. But the values do align with currently characterized sulfatases. The next part is going to be the structural analysis, and so I started optimizing crystallization of BFO2291. We discovered accidentally that it crystallizes in imidazole, and so I've done some work to improve the crystallization, but the best I was able to achieve was about a little over 3 angstroms, and the structure couldn't be solved with molecular replacement. BFO2290, on the other hand, I was able to crystallize despite how unstable the protein was, and it diffracted. And as you can see, it took several rounds of optimization, but eventually it crystallized in sodium citrate, ammonium sulfate, lithium sulfate at about 10 mg per mil. And I think the cryoprotectant we used was 30% solution of glycerol. We ended up with a data set at 2.85 angstroms, and Dr. Suits used molecular replacement to solve the phasing and found 3 proteins in the asymmetric unit. The statistics so far don't look too bad, but there is definitely room for improvements, especially with regards to the resolution. I also started crystallizing BFO2285, but I never got around to diffracting it before the whole COVID crisis. So with future directions for this project, both BFO2290 and 91 need improved crystal structure resolution, and especially with BFO2291, we can't solve with molecular replacement, so for phasing we're going to need to do selenomethionine crystals. I'm also hoping to use NMR and mass spec to determine the structure of the products for each of these enzymes, especially with BFO2285, where I need to get the protein active first. And for the overall Opteron system, I was hoping to use RT-PCR to find evidence for the upregulation of this operon by inducing a culture of Tannerella forsythia with chondroitin sulfate. Finally, at some point, I hope to do some cell localization studies for these proteins, because the server I was using, PSort, to predict the localization of the proteins was unable to determine where a lot of these proteins were localized. And with that, I'd like to thank my committee as well as all the members of the Suits Lab. Thanks for listening.